Hello and welcome back to Medical Compliance with Clarissa. I'm Clarissa Benfield, our Global Director and Business Line Leader for our medical and laboratory businesses here at Intertek. And today I'm very happy to introduce our subject matter expert and my guest, Tino from our Basel, Switzerland lab. Hi, Tino. How are you?
Good. Hello, Clarissa. Hello, everyone.
Yes, so happy to have you here with me today. So, Tino, what I always like to do to get us started is to have our subject matter experts introduce yourself a little bit. Tell everyone where you come from, what you do within the industry, how you got here, and then we will dive into our exciting topic for today.
Okay, I will do. My name is Tino Otte. I'm Director of Business Development at Intertek in Switzerland. We are a laboratory close to Basel. It's a border to France and Germany, and our lab is specialized in extractable leachables studies for medical devices, but also for pharmaceutical products. And we are dealing with this topic since nearly 20 years. I'm a polymer chemist by education, started at Intertek in 2016. As an expert for E&L studies and now serving a more commercial role. And before joining Intertek, I was working for several CROs in the pharmaceutical testing industry.
Yeah, wonderful. So you have a really extensive background. Obviously, you've been in this space for quite a while. This is a topic that's coming up more and more, both on the medical side and on the pharmaceutical side, talking about leachables and extractables and everything that manufacturers need to be doing. We see regulators thinking about this as well. So recently, kind of at the end of last year in the fall, right, September 2024, we saw that the FDA released a guidance document. You've put out some information regarding the guidance document. So maybe for our audience, you could tell them a little bit about what was this guidance document that the FDA released back in September? What was the goal that they were going after with the document? And really, why is it important? How does it relate to specific types of medical manufacturers?
Yes, so this guidance document is explaining a little bit more in detail how a biocompatibility study with focus on chemical characterization needs to look like. Obviously, there have been some dossiers submitted to FDA which were quite heterogeneous in quality and design. And for this reason, obviously, there was a need to explain a little bit more in detail what is really expected. So the ISO 10993/18 is an ISO chapter which describes the chemical characterization, and this guideline goes a little bit more in detail, and also it highlights several points which need to be addressed. That's the whole story behind it. It's like some points are really the same or repetition like it is written in the ISO 10993, and some topics are a little bit more sophisticated. So in some points, a little bit more needs to be done to be really compliant with the expectations of the FDA.
Yeah, and I think that's such a good point, right? We know these guidances, they're not just a nice to have, right? When the FDA puts them out, they really do expect that the industry is going to follow them, right? And they're saying exactly what they want to see. They're trying to be as clear and explicit as possible, right? So as you say, the guidance has the ISO 10993/18, but then there are these additional specifics more that's needed on top of just following the -18, right? So you have to go above and beyond that. Are there specific things that you notice in the document, or specific things that someone should look out for that's, above and beyond what -18 asks for that's listed in the document? Like, are there things that, are different that the FDA is expecting to see?
Yeah, so the -18 is, let's say, it leaves some space for scientific justification and flexibility. And the guidance now goes a little bit more into the detail. The biggest differences are on the characterization end. So it explains a little bit more how to do really the analytical testing. For instance, it really recommends to use a blank or control sample. It mentions that, for instance, cutting should be avoided as much as possible. And interesting aspect is also for the GC analysis, when you have to do a back extraction of maybe an aqueous solvent with an organic solvent. You have to do a spiking recovery and changing of the pH to ensure that also more polar compounds are properly transferred to the organic phase. Then it recommends sealed vessel extraction and mechanical agitation, so shaking or flow extraction. Then there should be a triplicate extraction of 3 batches of your material, which is also quite sophisticated and really a really hard requirement sometimes. And yeah, and there's again highlighted the general qualification and verification also for extractable screening methods, which is very similar to the requirement of a whole method validation. And the guideline really focus on high resolution mass spectrometry as a detector of choice. So everything should be done with an Orbitrap system or time of flight system wherever possible, because there's a very strict requirement for proper identification. So ideally for the tox assessment, all components should be confident and not just tentatively identified. So that's the majority of the difference.
Yeah, and I think it's really interesting to see too, right? I mean, that is above and beyond what the standard is calling for. And I think that just shows what the FDA is thinking, right? And how important this is, that they've outlined all of these things that they expect to see from manufacturers with these products that are going to be used on patients, right? They're coming in contact with the body, right? It's not maybe as simple or as straightforward as it was before, but they've learned with all of these products that, you need to provide all of this evidence and do all of this additional testing to really show that you've done your due diligence, knowing that these are going to be used on patients in the field everywhere, right? So I think it is really interesting when you look at the guidance in that way, seeing, yes, we can look at a standard, you can point to the 10993-18, but in addition, they see so many other areas where they want you to provide evidence and go even deeper, right? Not just one sample, but multiple samples, right? Being able to show that you've really done your due diligence and are taking all these things into consideration. I think too, right? So you talk about it a little bit with some of this additional testing. When someone's preparing their submission, obviously they need to think even further ahead, because some of this testing is not something you can do overnight, right? It's not something that you can just say, okay, we need these results right away, right? It takes some time. So when someone is thinking about their submission, it's not as simple as, oh, let's just throw this on the back end. You really need to be planning on how you're going to incorporate the requirements of this guidance into your overall testing process.
Yes, I would recommend to start very early, thinking about the chemical characterization, thinking about the new guidance, because the problem is it's really hard requirements. They need the high resolution mass spec here. And also keep in mind, many service providers will not be able to offer this service. And there could be like a big backlog of projects, and then it's difficult to get a slot on time. And also interpretation work could be quite sophisticated, and you should really, really think in detail about the design of the study and how to properly select the extraction solvents, extraction conditions, to be careful not to overestimate the extractables, because this could end up in a high number of compounds and maybe half of it could not be identified. So for this reason, it is very important to align the study really to the product, to its intended use, and also to what is already known about the materials and their compatibility with certain solvents.
Yeah, I think that's a really good point too. And when you talk about it, I mean, the chemical characterization, is there a basic way to explain what chemical characterization is, like what they mean when they say that, what they're looking for? When we use this phrase, right, we talk about it a lot. What is chemical characterization, and how does that apply, right, to a med manufacturer? What does that mean in general?
Yes, yeah. So there are different ways how to address the chemical characterization. And in the ISO norm, there is a flow scheme what could be done. And in the past, it was very often possible to do like a paper-based assessment using supplier data of your construction materials, to go through the different composition of the materials and maybe some of the additives. But the problem is, in most cases, this is never the complete picture because also the suppliers of the raw materials are sometimes not really aware about the impurities they have in their product. And the second point is the product or the raw materials will be processed, and then there could be also a change of the impurity profile. For this reason, chemical characterization means in principle an extractable screening, so an extraction ideally of the whole device, or in some cases it could be also a separate extraction of the construction materials, and then these extracts are subjected to different analytical techniques which are able to separate a huge variety of compounds from polar to non-polar, from volatile to non-volatile, and from organic to inorganic, to make sure you will not miss any compound or any unexpected impurity. And you need different separation and detection methods for doing this study properly. And normally there is a combination of GC-MS, LC-MS and ICP-MS coupled to different MS detectors to get the full picture of all the compounds. And there is like a quantification threshold, the so-called AET, analytical evaluation threshold, and you have to be able really to identify and quantify all the compounds which are detected above the AET concentration in your extraction solution, and then you can claim that if nothing is detected, you will be happy. But in most cases, you will see a lot of compounds. And once they exceed this level, they need a more detailed toxicological assessment where a certified toxicologist will do a structure specific prediction of the toxicology and comparing against literature data to ensure there is no concern associated with your device.
Yeah, and as you're going through this process, right, as like from the side of the manufacturer, right, as a manufacturer, I'm being told I need to flag all these materials and other potential risks and look at the worst case, right? Is that on me as the manufacturer to come up with the test plan, to come up with, what I should be doing? Is that where you engage with the lab and the lab makes recommendations, right? How do you approach this? Do you come to it with, okay, this is what we think as the manufacturer is going to demonstrate compliance? Or do we say, hey, this is our product, this is our use case, can you help us to understand, we start with step 1, move to step 2, move to step 3, right? What's the easiest way to address it, especially for manufacturers if they're going through it the first time and maybe don't have all of the expertise to know exactly what's required?
Yeah, so or the more common case is we have the final device produced by the manufacturer, and it needs to be qualified for biocompatibility. And then normally we recommend, depending on the use of the device and the different composition of the device, we recommend like a test plan, which is in agreement with the ISO 10993-18 and of course the new guideline. And then we will really discuss with the company which could be the most appropriate approach, which is covering the worst case requirement of the guideline and also of the ISO norm, but which is not overestimating or degrading the device. This is very important because when you design the extractables experiments in a good way, you can expect also good results in the analytical testing and identification. And normally, every manufacturer needs to be aware this is not the simple checkbox test anymore. It's really a dedicated analytical study where you could have situations where you have to adapt the study in the middle just to address some problems which may be only visible during the study itself. So it's really scientifically study here.
Mm-hmm. So when a manufacturer is going through this process, and if you look at the extractables, right, and you're going through it, and as you say, you may have to adjust your plan, you may have to change and do things differently as you're going along. Maybe it's slightly different from what the FDA is asking for in the guidance. Do you provide, as a manufacturer, do I then write a justification and say, okay, we started this and this is where we ended up? Or do you just follow the guidance as it is? Or how do you navigate that process, especially with the guidance being so new?
Yeah. Yeah. So it's this is a good point. So normally there are two ways to address problems. One problem could be maybe let's assume we have observed many peaks and some of the additives exceed the critical level, then one point could be to modify the production process or the raw materials. Or another point is to have another look on the extraction and to check if maybe the material was disintegrating or swelling or degrading in an overpronounced way. And this we can normally show on using the analytical data we have obtained after the extraction. And if you can show that the standard extraction condition recommended by the ISO or the guideline is not working, normally FDA always accepts deviations as long as you can explain that you can show or that it's still safe for the patient and it's still showing you what could worst case be administered to the patient. And that's very important.
Yeah, always important how you document it, right? How you document your justification, walk through that process. So it's very clear to an FDA reviewer. They understand, okay, this is how you got to where you are. This is how you're making sure that your product is safe and effective, right? So always about how you're justifying it and document it, of course, is really important when you're dealing with the FDA or any regulator in general, right? So the other thing you mentioned, Tino, which I think is interesting and maybe we can circle back to is, when you're talking about the material variability, some of the batch testing, right? You have to test across 3 different batches. Mm-hmm. So is this different than what was required before? Why is it important to test across 3 batches, right? What does this mean as a manufacturer? Like, are there challenges with that when you're looking at, now testing across 3 different batches of materials?
Yeah. So I have to say, there is also like an extractable leachables world in the pharma space. And I have to say now with the guideline, the requirements are quite similar like they are already for pharma contact materials.
Of course.
That's one point here. And I think also in pharma, the FDA is desperate for multi-batch testing. Mostly in the pharma world, it's the formulation which needs to be tested for different batches. But here we are talking about a medical device, and there, of course, it's the material. So we should really test 3 different batches of the material. And this is really well justified because for instance, for a plastic material, you can expect a much bigger variability batch to batch, or even within the same batch, you can see differences because it's not so well defined like a pharmaceutical formulation. Mm-hmm. You have many impurities where the production company is not aware of, and sometimes raw materials are exchanged against raw material from a different supplier with the same specification, but unfortunately, these polymer specifications normally do not cover impurities. And when we do such an extractable study or chemical characterization, we detect at very low concentration, and then we will see any difference in overpronounced extent. And this is always a nightmare for polymer. You can expect really different results. And for this reason, I believe personally, it's also scientifically well justified to get at least a good idea about the variability in the polymer. And then I think this will remain mandatory in future for medical devices.
So we say 3 batches. Do you ever see people wanting to test more than 3 batches based on the risk of their products or based on their supplier? Or is 3 pretty standard, or we see people go above and beyond that requirement?
Yes, that's possible. Sometimes it really depends where you are in your development process. Yeah. If it's just, it depends if this is really commercial batch already or if it's just prototype batch. If it's prototype, you should really test more because variability is expected to be more variable. Yeah. And this is very important to know. And for some devices, when we are talking about high risk, if there are medical devices which are used somehow in context with some inhalation, then you have a very high risk and very low thresholds due to the direct exposure of potential leachables to the lung cells. And then I know there are a lot of companies, they are doing a 100% batch control anyway. For extractables. So everything is possible.
So kind of across the board. Yeah. Interesting. Yeah. The other thing that they mention in the guidance document is this analytical evaluation threshold, the AET. So can you talk a little bit about that? It's kind of unclear in the guidance. What does that exactly mean when they talk about this threshold?
Yeah, so it's the AET principle. As a kind of definition, maybe the AET can be regarded as like a critical concentration in our extract. And below this concentration value, any detected compound can be regarded to have no tox effect independently from its structure because the concentration is that low. Yeah. But above this threshold, all the compounds need to be structure elucidated. So you need to have a structure because a toxicologist needs to do a structure-specific toxicity assessment. And also you need a proper quantification because the real concentration plays also a role for the tox assessment performed. Yeah, and quantification is done like a bottom line approach. So you will have your AET level and everything above it needs to be really characterized in detail. The AET principle is valid for many compounds. But it's some compounds of cohort of concern, like for instance, nitrosamines are excluded from the AET principle because they are known to be highly toxic and they have even lower threshold. Yeah. And calculation of AET, there is a formula given in the ISO, which is not too complicated, so to say. It's in principle, it's a kind of dose-based threshold. So it considers a kind of safety threshold, which can be taken from several sources. For instance, one source could be the PQRI guideline, for parenterals, or there would be, for instance, a safety concern threshold of 1.5 microgram per day for a parenteral product, which would be also applicable, for instance, for implantable devices or similar cases. Or you can anticipate a safety threshold from the ICH M7 guideline for genotoxic impurities. And there, the TTC threshold of toxicological concern concept is applied. And there you can even justify higher values if there is a limited in lifetime exposure to your device. So if it's just once used in lifetime or at least limited time in lifetime, you can justify higher values. And then you have just to convert this microgram per day value into a concentration because we are testing extracts here. And this means you have to, into the formula, you have to consider the volume of your extracts, the number of devices extracted, and of course, the number of devices exposed to the patient per day. This is the dose part of the formula. And you have to consider an uncertainty factor, which again depends on your analytical method you are using. Because when we do a screening, we do a semi-quantification where we, in most cases, will use a high number of different characterization standards. And then we use the average response factor to determine the concentration of our unexpected extractable or unexpected leachable. And depending on the technique, there could be a relatively high variability of this response factor. Yeah, and for GC, MS or also GCFID, it's okay. There's a low variability and there the uncertainty factor is around 2. But when it comes to LCMS, it's more difficult because you could easily have uncertainty factors of 10 and higher, which means there is a huge dependency of your detector response on the concentration, which depends again on the structure of your compound. And then it's sometimes better to use the LOQ of your instrument instead of the AET. For detection of the peaks, and then you have to do additional work to characterize your peaks and to use an absolute quantification where you inject a chemically related standard to get a proper response factor for the LCMS. So it's not the principle of AET is simple, but how to apply it in the study is quite complicated in some cases.
Yeah, exactly right. How you get there can be a little bit more challenging, right? Yeah. So simple concept, and then getting there can be a little bit more challenging. Well, Tino, before we go, I want to chat a little bit too. Obviously, biocompatibility, this is a huge topic. It has been in the industry for a while, right? We saw the guidance come out in September. We know that the FDA was taking feedback, as they always do, through to December, right, of 2024. Is there anything that, you think from an industry perspective or things that you're hearing? I know, again, biocompatibility is a topic that comes up a lot. Everybody has opinions on it, on how the testing should be done. There's just a lot of comments and discussion in the industry. So is there anything that you see on the horizon, right, with the guidance or just in general with how biocompatibility is going to be applied, or new ways to show biocompatibility, right, that are coming out? Anything you want to share with our audience before we go?
Yeah. As a general point, there will be a revision of the 10993-1, so the basic chapter of the biocompatibility. And if we focus on the chemical characterization, I would expect, since the FDA published this draft guidance, obviously this draft guidance addresses all the points which have been of concern for the FDA in the past. So I would expect that we will see maybe a final version of the draft guidance very soon, which probably will not differ a lot from the current version. So I would even expect that it could be even worse from a kind of study requirement point of view. And maybe then there could also be a revision of the chapter 18, which considers the guidance a little bit more. That's possible. And since all is still guideline, you can also refer to other E&L guidelines, for instance, as they are used in the pharma area. There's the USB 1663, 1664, and there will be in future like an ICH guideline, the ICH Q3E, which tries to harmonize all the E&L testing, at least for the pharma world. And I think this could also be a good reference, which could be used to address some of the not well explained parts in the ISO 1099318 and also in the guideline a little bit more in detail, because it's a worldwide recognized standard, the ICH, and could be also a good addition to the current guideline for the medical devices.
Yeah, absolutely. Well, Tino, there's a lot to unpack. There's such great information. So glad you could come on and join us today. Again, we have a lot of other content out there that you've created based on these topics. So if anyone is interested or has questions, as always, they can reach out to me directly. I'm happy to connect you with Tino and his team. Thank you so much for joining, and we'll chat with you guys next time.
Thanks a lot.